well? This is because your sense of taste is sensed by your taste buds, which, when damaged, do not taste the food you eat.
No.
Now let's look at a diagram of the tongue. Your tongue has 5 types of taste buds that taste sweet, sour, salty, bitter, and umami. Umami is the taste of savory things such as chicken or steak. Contrary to common belief, these fine tastes are not necessarily grouped in certain areas of your mouth. The different taste receptors, as seen on the screen, are spread out in your mouth, which is why a person without a tongue can still taste things. Taste or gustation is a chemical taste. Chemicals from food interact with gustatory cells inside taste buds to create neural impulses which are sent to your brain. As seen over here, this is the taste bore, which is pretty much the taste bud, and these are the taste receptor cells, which are called basal cells. Taste and smell are indirectly connected. Have you ever noticed when you smell something delicious like freshly baked bread, tastes better, or vice versa with bad smell? This is because there is sensory interaction between taste and smell. Smell or olfaction, like taste, is a chemical taste. When you smell, part of what you are smelling enters your nose. These are called odorants. These odorants enter into the nasal cavity. That's the nasal cavity. This odorant chemical stimulates 5 million receptors that send a nerve impulse to your olfactory bulb in your brain. This is a picture of the brain. The olfactory bulb, which receives the nerve impulses from your receptors in your nose, is closely connected to your limbic system. So here is the olfactory bulb, or around this area, and it connects to the hippocampus, which is around this area, which is why memories can be stimulated by your senses of smell. This is because hippocampus is in charge of memory and navigation.
The most noticeable and outward portion of the ear is the outer ear. The fleshy outer ear is called the pinna, or specifically for humans, the auricle, leading to the ear canal and to the outer tympanic membrane, or the eardrum. The middle ear is in an air-filled cavity behind the eardrum and includes 3 bones: the malleus, or the hammer, the incus, or the anvil, and the stapes, or the stirrup, altogether known as the ossicles. The ossicles act as a bridge for sound, allowing the sound waves to pass through the eardrum from a series of vibrations. The middle ear is essential in the transportation of sounds throughout the ear. Beyond the middle ear, further in, is your inner ear. The inner ear is a bony labyrinth containing sensory organs that assist motion and balance. Such organs include the vestibules of the ear, the utricle and the saccule, and the sensory organ, the cochlea, which allows for hearing. The cochlea is composed of 3 fluid-filled spaces: the scala tympani, the scala vestibuli, and the scala media, all responsible for your hearing. The way we hear is through sound waves traveling through the outer ear and processing in the middle ear, which is transmitted through the vestibular cochlear nerve in the inner ear. This nerve pushes information directly to the temporal lobe of the brain, registered to us as sound. Another central function of the ear is providing balance. The ear creates 2 types of balance: static balance, which allows people to feel gravity, and dynamic balance, which allows people to sense acceleration. Deafness or hearing loss may be caused by a number of factors, including aging, genetics, noise exposure, infections, birth complications, ear trauma, or medications and toxins. A common cause is chronic ear infection. Hearing loss is typically diagnosed if a person cannot hear 25 decibels in one ear during a hearing test.
Also related to sound are sound waves. Sound waves travel through the air through compression and refraction. The frequency of a sound is the number of waves per certain amount of time. The higher the frequency, the more amount of waves. This is perceived as highness or lowness of sound, which is called pitch. The higher the pitch, the squeakier you sound. Amplitude is the amount of energy in a wave. Amplitude is measured by how tall or how short the wave is. The higher the wave, the louder you sound. The softer you sound, the lower the wave. That is called intensity, which is the volume of sound. Also determining sound is the quality, which is timbre. These are the characteristics of a sound. So when you hear something, it may be different because of the quality or timbre of the sound.
This right here is a picture of a labeled eye. When light hits our eyes, the first part to take action is our cornea. The cornea is a transparent tissue that covers the front portion of the eyes. This protects the eye and bends light to provide focus. Once the light enters through the cornea, it passes through the pupil. The pupil is an adjustable opening in the center of the eye through which the light enters. A ring of muscle that forms the colored portion of the eye, called the iris, works with the pupil to regulate the amount of light entering the eye. Through dilating or constricting, the iris can adjust to the light and also react to inner emotions. For example, when we feel amorous, our pupils will dilate. Past the pupil, the light is received by the lens. The lens changes its curvature in order to focus incoming rays into an image in a process called accommodation. When the light rays have passed the lens, it will then reach the retina. The retina is a light-sensitive inner surface of the eye that contains sensory receptors that process visual information and sends it to the brain. When the light reaches the retina, because it was moving in a straight line, it will cause the image to be upside down. However, this is not a problem because our retina does not look at the image as a whole. Instead, the receptor cells convert the light energy to neural impulses that get sent to the brain, and in the brain will be where the information gets organized and perceived. By the center of the retina is the fovea. The fovea is a focal point and provides sharp central vision. Receptor cells called cones are clustered here, allowing this area to better detect fine detail. At times, when there is an abnormality and the focus is not in the fovea, vision problems occur. Nearsightedness is a vision problem that occurs when the lens focus is in front of the fovea. This causes perception of near objects to look clearer than distant objects. In extreme cases, an individual will not be able to see anything clearly. However, this problem can be fixed or adjusted through glasses, contact lens, or LASIK. Opposite to nearsightedness, farsightedness occurs when the lens focus is behind the fovea. This causes faraway objects to see more clearly. At a young age, this is not much of a problem because of the ability to accommodate, so glasses are rarely needed. Under the retina's outer layer are the receptor cells called rods and cones. The cones are concentrated near the center of the retina and function to have low sensitivity in dim light and be color and detail sensitive. Contrastingly, the rods function to have high sensitivity in dim light and are not color and detail sensitive, meaning cones are good for daylight and well-lit areas and rods are good for peripheral and twilight vision when the cones are not at work. How the system works. When light strikes the receptor cells, they produce a chemical change that generates neural signals. These signals would activate the neighboring bipolar cells, which then activates the neighboring ganglion cells. The axon from the ganglion cells would converge to form an optic nerve that would carry the information to the brain. Now that we talked about the visual process from light, let's talk about the light and its colors. How do we see color? Our eyes are not hit by color itself. Instead, pulses of electromagnetic energy strikes our eyes and allows us to experience color. In the opponent process theory, Herring theorized that we process 4 primary colors combined in pairs: red, green, blue, yellow, and black, white. These opposing retinal processes enables colored vision. We see color, but how should we describe our experience of it? We are able to determine or experience color through its physical characteristics: wavelength, hue, intensity, and saturation. Hue is a term we give for colors we experience, such as blue or green. Hues are determined by the light's distance from one wave peak to another, called wavelength. Intensity is the amount of energy in light waves and is determined by the wave's amplitude, or in other words, height. Intensity often influences the brightness, where big amplitudes mean bright colors and small amplitudes mean dull colors. Saturation is the ratio of dominant wavelength to wavelengths in the color and points out the purity of a color. If a saturation is high, the color is rich, and if it is low, the color is dull. Light to color, image to eye to brain. Our eyes work in these miraculous ways to give us vision every day.
Touch, also known as the somatosensory system, is often one of the most commonly forgotten senses. It is the first sense developed when we are born that connects us to the complex external world by bringing in information from receptors in the skin and sending information to the brain that we register as sensation. These receptors, located all over the body, are responsible for detecting these specific sensations, which are pressure, warmth, cold, and pain. In order to register these sensations, there are three types of receptors. The first being the mechanoreceptors that are in charge of detecting pressure, texture, and vibrations. In the skin, they are known as Merkel's discs and Meissner's, Ruffini's, and Pansinian corpuscles.
Their only function is to perceive pressure and indentions on the skin. It has been found that Merkel's discs and Meissner's corpuscles, the most sensitive mechanoreceptors in the skin, are located on the parts that do not have hair, on the top layers of the epidermis and dermis. These sensitive receptors are located on the tips of our fingers and the palms of our hands, along with the soles of our feet and other areas, that allow us to gather large amounts of information about the texture of objects. This could explain why most people can gather more information about the way something feels by touching it with their fingertips rather than the side of their arm. Ruffini's and Pacinian's corpuscles are more used to detect vibrations of the skin and move down bones and tendons, like the movement of limbs and stretching of your skin. For temperature, thermoreceptors are used, which are located on the dermis layer of your skin. These are made to perceive the temperature of your skin at any given moment. The two categories for these receptors are hot and cold. Cold receptors are activated when the temperature of your skin reaches below its normal temperature, which is usually around 95 degrees. They continue to be stimulated until the skin reaches all the way down to 41 degrees. Beyond that point is when you lose sensation and you go numb. Hot sensations are perceived when the skin reaches over 86 degrees and is the most stimulated at 113 degrees. When your skin reaches beyond this temperature, the pain receptors begin to take over to prevent the damage of tissue. Both of these receptors are located all over your body, commonly around your face and ears, which is where you get red first when standing outside on a chilly fall morning.
And now to the pain, which despite its uncomfortable feeling, is one of the skin's most important senses. Pain receptors, also known as nociceptors, is basically the body's defense mechanism to protect yourself and to maintain homeostasis. It's the body's way of telling you that something is wrong. In total, throughout your body, there are over 3 million nociceptors in the skin, in muscles, in blood vessels, and even in organs. That kind of gives you an idea of how important it is to feel pain every once in a while. But before you feel the pain, there's a quick process that your body goes through.
First would be the stimulus, and in the example of getting a paper cut, the stimulus would be the direct contact of the paper tearing the skin. Immediately after, free nerve endings, as shown on the screen above, sense this change. Free nerve endings are located on the top epidermis layer of your skin. Once these free nerve endings gather the information is then sent to the central nervous system and onto the brain where pain is created. This feeling of sharp pain that the brain creates alerts you to move immediately away from the stimulus that's causing pain. After a while, the sharp pain turns dull and alerts you not to touch the damaged area while it heals. It's important to remember that the brain creates pain after receiving information, not the other way around.
Related to this concept of pain is the gate control theory. The theory that the spinal cord contains a neurological gate that blocks pain signals or allows them to pass on to the brain. During normal somatosensory activity, there are inhibitory and projection neurons. However, the inhibitory neurons prevent the projection neurons from sending signals to the brain.
Hence, the gate is closed. The gate is opened when there are only small fiber stimulations and inhibitory neurons are deactivated. This allows projection neurons to reach the brain, signaling pain.
While we do have 5 main senses, we also have several underlying ones that help us in everyday life, like our vestibular sense. Our vestibular sense is our ability to control our spatial coordination and our sense of balance. Their main network is located in the inner ear in the semicircular canals. These canals connect the vestibular sacs to the cochlea. When we move or tilt our head, hair-like receptors in the inner ear send information to the cerebellum, where we get a sense of our sense of position and our balance.